if you were to hire an architect to build 4 completely different structures.
Okay, like what kind of structures?
Like, let's say, a sprawling underground subway network, a highly fortified military bunker, a chemical processing plant, and, I don't know, a massive air filtration center.
Right, 4 very different jobs.
Exactly.
Yeah.
And if you handed them the exact same pile of generic bricks and just one single blueprint, they'd look at you like you had completely lost your mind.
Oh, absolutely, they would. I mean, and rightfully so, because in engineering, the raw materials really have to match the functional demands of the structure.
Yeah.
You don't build a delicate filtration system out of the same heavy-duty materials you use for a bunker.
Right. But today, we're looking at a biological marvel where your body does exactly that. And we're talking about something that happens inside of you literally every single day.
It's wild to think about.
It really is. You've probably heard the gut referred to as your second brain. But what completely blew my mind in our research for this deep dive is that your heart, your lungs, and your pancreas, they actually have their own mini brains too.
Yeah, it's a fascinating area of biology. They are officially called organ intrinsic nervous systems, or OINS for short.
OINS, got it.
And these aren't just passive wires receiving orders from your actual brain in your skull. They are embedded, local neural circuits that actively shape the physiology and function of our vital organs.
And to figure out how this works, we are diving into a massive stack of recent research today, anchored by a really groundbreaking 2026 paper published in the journal Nature.
Right, the paper by Sue and colleagues.
Yes, Sue et al. So our mission for this deep dive is to answer that architectural mystery. How do these completely different mini-brains, with radically different shapes and daily jobs, all get built from the exact same starting material in the embryo?
It is a profound question, and the researchers uncovered a brilliant dual logic to explain it.
A dual logic.
Yeah, so what we're going to explore is this stunning interplay between a cell's ancestral history, like where it came from, and the surprisingly aggressive influence of its new neighborhood.
Well, let's start at the very beginning then. Where do all these mini-brains actually come from? If they share a starting line, where is it?
So they all originate from one very specific embryonic source. They are called the vagal neural crest cells.
Okay, vagal neural crest cells.
Right. And to be precise, they come from regions along the early spinal cord known as somites 1 through 7.
Somites. Let's define that really quickly.
Good idea. You can think of somites as, well, early developmental blocks in the embryo, just foundational units.
So from the single origin point, these crest cells just migrate out into the developing body to populate the organs.
Exactly. From those somites, they pack their bags and head out.
Okay, so they all start in that early spinal cord area. But they obviously don't all travel the exact same way, right? Because the organs are in completely different physical places.
That's the first major divergence we see in the study. The research highlights 2 radically distinct migratory routes.
And those routes basically prefigure the physical layout of these mini-brains.
Yes, precisely. The first route creates the enteric or gut nervous system, as well as the pancreatic neurons.
Okay, the gut and the pancreas.
Right. And these come from what we call enteric neural crest cells, or ENCCs.
ENCCs.
Yep. And by embryonic day 11.5, which researchers call E11.5, these cells form visible, long migratory chains.
So they're physically linking up and moving.
Exactly. They spread out incredibly widely, covering just a massive amount of physical territory across the entire digestive tract.
It almost sounds like they are casting a massive net, like explorers spreading out across a vast new continent, just trying to cover as much ground as possible.
That's a really helpful way to visualize it, but the second route is entirely different.
How so?
The neurons destined for the heart and lungs, they don't spread out at all. They derive from Schwann cell precursors, or SCPs.
Okay, SCPs, and they travel along the vagus nerve, right?
Yes. By embryonic day 12.5, or E12.5, they enter the heart and lungs. But instead of migrating everywhere like the gut explorers, they drop into discrete, concentrated sites.
Like where?
Like the atrial center of the heart, or the hilum of the lung, and they just stop. They stay there, forming these dense, compact clusters called ganglia.
So if the gut and pancreas neurons are like the explorers spreading a wide net, the heart and lung neurons are more like paratroopers dropping onto a single, highly strategic base and aggressively digging in.
I like that analogy, paratroopers.
Yeah, they don't want to spread out, they want to fortify one spot.
Exactly, but the big question the researchers had was why they behave so differently.
Right, because if they all start from the same vagal neural crest starting line.
Mm-hmm.
What is the biological mechanism that tells the explorers to keep moving, but tells the paratroopers to stop and set up camp?
Yeah, how does a microscopic cell know it's supposed to start moving? It doesn't have a GPS.
Right. So what is the mechanism?
It actually comes down to an internal genetic toggle switch.
A toggle switch.
Yeah. In those gut and pancreas explorers, the cells are proliferating, sure, but they heavily express a gene called SOX10.
SOX10.
SOX10. Yes.
Okay.
You can think of SOX10 as a biological engine. It drives their migratory capacity. As long as SOX10 is turned on, the cells keep moving.
Like gas in a car.
Exactly. And simultaneously, they express a master regulator gene called PHOX2B.
PHOX2B. What does that one do?
It essentially assigns them their role. It's like an identity card saying, you are destined to be part of the organ intrinsic nervous system.
So they have an identity card, PHOX2B, and a running engine, SOX10.
Right.
But what about the paratroopers in the heart and lungs?
They drop the SOX10 completely. Very early on, they just turn that biological engine off.
Oh, wow. They just kill the engine.
They do. They keep their PHOX2B identity card, but they replace the migratory engine with markers that indicate active rapid cell division, like a marker called Chi67.
Wait, Chi67.
Yeah, it basically means they become non-migratory neural progenitor cells, or NPCs.
So instead of moving, they just multiply.
Precisely. They lose their migratory potential entirely. They multiply rapidly to build a local dense population, but they stay exactly where they landed.
Okay, let me just make sure I'm wrapping my head around this. Essentially, these cells all start in the exact same place in the embryo.
Symphonites 1 through 7.
Right, but their physical layout, whether they end up as a sprawling net of wires in the gut or a dense, anchored clump in the heart, that is entirely dictated by their travel route and whether they leave their migratory engine, SOX10, turned on or off.
That is the foundation of the spatial map. Lineage determines the entry route, and the genetics dictate the spreading versus the clumping.
It really makes you think about the mechanics of your own body. If you listening right now were designing a heart, which is a muscle that violently beats like 100,000 times a day, the last thing you'd want are loose, delicate neural wires scattered everywhere.
Right, they get destroyed.
Exactly. You'd want tightly anchored relay stations so they don't get physically torn apart.
It's a very elegant structural solution to a massive mechanical problem.
Okay, so let's do a quick rapid-fire recap for you listening, just to summarize everything from this first part of our deep dive.
Good idea. Lock in those concepts.
Bullet point 1. All visceral mini-brains, the OINs, start from the exact same vagal neural crest cells.
Bullet point 2. Gut and pancreas neurons act like a spreading net. They are SOX10 positive and highly migratory.
Bullet point 3. Heart and lung neurons act like anchored clumps. They lose SOX10 and stay localized as paratroopers.
And bullet point 4. The spatial layout is entirely prefigured by the cell's lineage and migration route.
Perfect. But having a map is only half the battle.
Right.
Which sets up the next crucial question for the researchers.
Right. If a cell's lineage explains where it goes and how it physically structures itself, does that lineage also explain its identity?
Like, does a cell's origin dictate what kind of neuron it ultimately grows up to be?
Exactly.
And it seems logical that it would, right? I mean, if you come from a certain family, you share their traits.
You'd think so.
But the plot twist in this research is that lineage absolutely does not equal destiny.
Not at all. And to figure this out, the researchers used a really advanced technique called single-cell RNA sequencing.
Let's translate that a bit, because it's kind of a dense term. My understanding is that it's essentially like reading the private diary of a single microscopic cell to see exactly which genes are turned on and off at a specific moment in time.
That really captures the essence of it, yes. But imagine trying to read thousands of these diaries simultaneously. They sequence these cells at embryonic day 14.5, so E14.5.
Okay, so slightly older embryos now.
Right, and they specifically tracked the cells with that PHOX2B identity card, looking at their transcriptomic trajectories.
Basically watching how they mature over time.
Exactly. And the twist was profound. The immature precursors looked almost entirely identical, regardless of which of the 4 organs they were in.
Wait, really? So a baby neuron in the heart looks exactly like a baby neuron in the gut?
Indistinguishable, transcriptomically speaking. But as they mature into final adult neurons, their molecular identities radically diverge. Their genetic diaries start looking completely different.
And this brings us to what I couldn't help but call the pancreas betrayal when I was reading our notes.
The pancreas betrayal, I like that.
Because remember, Pancreatic neurons come from the exact same explorer lineage as the gut neurons.
The ENCCs.
Yes, they travel the same way. They keep their SOX10 engines on. They are literal developmental siblings.
They are.
But when the scientists looked at the mature cells, the pancreas neurons ended up looking much more like heart and lung neurons than their own gut siblings.
It's a fascinating divergence. The transcriptomic data showed that despite their shared history, the gut neurons split off into their own highly distinct developmental branches.
And those branches are defined by specific transcription factors, right? Like ETV1 and BNC.
Exactly. Gut neurons get marked by ETV1 and BNC2. Meanwhile, the pancreas neurons abandon that path entirely.
The betrayal.
Right. They share completely different developmental branches with the heart and lung neurons. And those branches are marked by different factors, specifically OIL1 and DL5.
Okay, so gut gets ETV1 and BNC2, but pancreas, heart, and lung share ISL1 and DL5.
You've got it. It proved definitively that lineage did not dictate their final identity.
This is where the implications get really wild to me. Because if the pancreas neurons share a history and a travel style with the gut, but they end up with a final molecular identity that matches the heart, does that mean the pancreas itself is somehow brainwashing the neurons once they arrive?
Well, brainwashing is a very human concept, but biologically speaking, yes. The local environment of the organ actively rewrites the programming of the cell.
Oh, wow.
And the researchers proved this definitively using in vitro co-cultures. They essentially recreated this interaction in a petri dish.
I read about this. They actually took developing gut nervous system cells, cells that were fully destined to become gut neurons, and they put them in a dish with heart cells or lung cells.
They did. And to take it a step further, in some tests, they didn't even use the heart cells themselves.
What did they use?
They just used the liquid medium that those organs had previously been growing in.
Like the juice.
Basically. The soup, if you will, full of the chemical secretions from the heart or the lungs.
And what happened to the gut neurons in that soup?
They were actively reprogrammed. Their private genetic diaries were rewritten. They shifted their transcriptional profiles to match heart or lung neurons.
So the organ's local environment was actively overriding the cell's ancestral lineage.
Exactly. Nurture overpowers nature in this case.
So just taking a step back here, while a neuron's physical location and its spreading pattern are set by its lineage map, its final molecular identity, its actual personality, is entirely instructed by the neighborhood it lands in.
That's a perfect summary.
It makes you wonder about the broader nature versus nurture debate we have about ourselves. Think about this for a second. This is the ultimate cellular nature versus nurture.
It really is.
How much of your own identity do you feel was pre-programmed at the start versus shaped by the environment you landed in? It's kind of wild to think our cells go through the same crisis.
And they adapt brilliantly to their new homes.
Okay, so let's hit our second quick recap for you guys listening. A quick 10-minute checkpoint.
Let's do it.
First, lineage does not equal destiny when it comes to a neuron's identity.
Second, single-cell RNA sequencing shows that immature neurons in all organs look the exact same.
Third, the pancreas neurons share a history with the gut, but they adopt an identity closer to the heart, labeled by those IL-1 and DL5 markers.
And fourth, organ-derived cues actively reprogram these neurons upon arrival, as shown by the Petri dish soup experiments.
Awesome. But this raises a massive mechanical question for me.
Which is?
If the pancreas or the heart is instructing these baby neurons on how to grow up, what is the actual physical mechanism? How does a heart cell talk to a nerve cell?
This is perhaps the most paradigm-shifting part of the whole CSoup paper. The secret architect behind all of this reprogramming is something called the extracellular matrix.
The ECM.
Yes.
The ECM. Now, historically, hasn't the extracellular matrix just been viewed as biological glue? Like the mortar holding the cellular bricks together so we don't just melt into a puddle?
That was the traditional textbook view, yes. Just passive structural support.
Right.
But this study completely flips that notion on its head. The ECM is not just passive mortar. It is a highly active, incredibly complex signaling hub.
How did they figure that out? Because looking at glue and realizing it's actually a communication network sounds like a massive leap.
It was a huge leap. To figure this out, the researchers focused heavily on the intrinsic cardiac nervous system, or ICNS.
The miniature brain inside the heart.
Exactly.
Yep.
And they used a tool called spatial transcriptomics, specifically a platform called Visium.
Visium, okay, so if single-cell sequencing is like reading a cell's private diary, what is Visium?
Think of spatial transcriptomics like taking a GPS map of a city and overlaying exactly where every specific conversation is happening.
Oh, cool.
Yeah, it shows you the physical location of gene activity within the actual tissue architecture.
And when they laid that GPS map over the heart, what did they see?
They saw that the ICNS sits perfectly, snugly wedged between cardiac fibroblasts and cardiomyocytes.
Cardiomyocytes being the heart muscle cells.
Right, and those surrounding heart cells are constantly pumping out specific extracellular matrix proteins, things like collagens and fibronectin.
So the heart is churning out this glue. But how is the glue talking to the neurons?
Through highly specific chemical pathways, the researchers used algorithms to predict cell-to-cell communication.
Like wiretapping the conversations.
Kind of. The algorithm they used is called cell chat, and it revealed massive amounts of ECM receptor signaling pointing directly from the heart cells to the neurons.
Wow.
The heart cells were talking via the matrix proteins, and the neurons had receptors listening to them.
But, science requires proof. They needed a smoking gun to prove the neurons were actually relying on those signals to grow up, right?
They did. So they decided to break the neurons' ability to listen.
How do you do that?
They genetically knocked out a gene in the neurons called ITGA1, integrin alpha-1.
Integrin alpha-1.
Yes. This gene is responsible for building a crucial chemical antenna, a receptor, on the surface of the neuron. Without it, the neuron is deaf to the ECM signals.
And what happened when they broke the antenna?
It was catastrophic for the neurons.
Really?
Yeah. They completely failed to differentiate properly into mature heart neurons, but beyond that, their tightly clustered, highly organized architectural ganglionic shape just fell apart.
Oh, wow. So they didn't just fail to mature, they physically disbanded.
Yes. They needed the signal just to hold their structure together. And to understand the physical breakdown further, the researchers looked at another gene called LOX. LOX.
I remember this one from the sources. LOX is responsible for cross-linking the extracellular matrix, right?
Exactly.
It essentially weaves all those loose fibers together to make them strong and resilient.
Right. Because the heart is a violently chaotic environment, it's constantly moving, contracting, and stretching.
Yeah, beating 100,000 times a day.
Exactly. When the researchers disabled the LOX gene, preventing that strong cross-linking, the ganglionic structure of the mini-brain couldn't withstand the mechanical stress of the heartbeat.
It just got torn apart.
It literally disintegrated under the pressure.
Okay, this is wild. Let's try to visualize this for everyone. Imagine the extracellular matrix in the heart is like a smart Velcro mattress.
Smart Velcro mattress. I like where this is going.
Okay, so it physically sticks the neurons in place, anchoring them so they aren't violently shaken loose by the heartbeat. That's the Velcro part.
Right, the LOX cross-linking, keeping it strong.
But it's a smart mattress because it's packed with chemical sensors, those collagens and fibronectins, that are constantly pumping instructions into the neurons through the ITGA1 antennas.
Telling them exactly how to grow up and specialize for the heart's specific needs, driving neurogenesis.
Driving neurogenesis, yes.
That's a highly effective way to think about it. The physical anchoring and the chemical signaling are fundamentally intertwined. You really can't have one without the other.
So, bringing this all together, we used to think the brain in our skull was the absolute dictator, just sending long wires down into passive organs to tell them what to do.
The old top-down view.
Exactly. But this research proves that our organs are actively building their own customized control centers.
The organs are co-constructors of the nervous system.
The extracellular matrix provides both the physical anchor to survive the extreme environment and the chemical instructions to specialize for it.
It's just phenomenal bioengineering.
Time for our final 15-minute rapid-fire recap. Let's lock in this last segment.
Okay, bullet point 1. The extracellular matrix, or ECM, is the communication bridge between the organ and the nervous system.
Bullet point 2. Specific genes like ITGA1, which builds in the antenna, and LOX, which weaves the Velcro, are required to read and survive these ECM signals.
And bullet point 3. Without ECM signaling, neurons completely fail to mature and fail to form their distinct protective clusters.
Which brings us to the ultimate synthesis of our deep dive today.
The dual logic we talked about at the very beginning.
Right. Lineage, the ancestral history of the cell, prefigures the spatial framework. It's the scaffolding. It determines if the cells cast a wide net like the gut explorers or drop in as clumped paratroopers like the heart neurons.
But the organ itself, using organ-specific cues from the extracellular matrix, acts as a programmer, providing the final molecular identity.
Scaffolding and programming working together in perfect harmony.
And this isn't just an abstract biological curiosity. Knowing how these OINS are built matters immensely.
Why is that? Why should everyone listening care about how their mini-brains are built?
Because these intrinsic networks are what sustain your whole body homeostasis. They govern your automatic functions, your digestion, your breathing rate, your heart rhythm.
Which naturally leads to the dark side of this equation. What happens when these mini-brains break down?
That is exactly the frontier researchers are racing toward right now. Disruptions in these organ intrinsic networks are increasingly being linked to severe autonomic dysfunctions.
Like what?
Things like immune disorders. But most surprisingly, they are being implicated in devastating neurodegenerative diseases like Parkinson's and Alzheimer's.
Wait, really? Parkinson's?
Yes. There is growing evidence that the breakdown of these networks plays a crucial role.
Wow. Think back to our opening metaphor, guys.
Yeah.
The architect, the blueprints, the bricks. We've realized today that the building site itself, the soil, the neighborhood, the extracellular matrix, changes the very nature of the bricks being laid.
It dictates their survival and their entire function.
So I want to leave you listening with a final provocative thought to mull over.
Lay it on them.
We spent decades trying to cure diseases like Parkinson's by looking almost exclusively inside the skull, right? Treating the brain as this isolated fortress.
It's been the standard approach, yeah.
But if these systemic diseases involve a catastrophic breakdown of this body-brain axis, what if the cure isn't found by looking inside the skull at all? What if the key to saving the mind is actually found by looking at how the gut or the heart's extracellular matrix is talking to its own intrinsic mini-brain?
If the soil of the organ is toxic, the neural network it supports will inevitably fail. It changes where we need to look for therapies entirely.
It really does. It flips everything we thought we knew completely upside down. Next time you feel your heart racing, think about the microscopic smart Velcro holding your cardiac mini-brain in place. Isn't it wild that your heart essentially built its own pacemaker?
It's an amazing machine.
It really is. Well, until next time, keep diving deep.